cells was a developmental biology term, but for stem cell biologists, we just say a liver stem cell and a lung stem cell, things like that. But the most remarkable thing is actually the fact that although this process cannot be stopped, and in the 80s, the late 70s and 80s, and scientists began, mostly in England, try to stop this process, at least in vitro, okay? And try to isolate embryonic stem cells by taking the inner cell mass from the blastocysts, try to grow them, namely arrest their development in the undifferentiated stage. And the advantage of that being, so in that, if you are successful, then you will be able to have a copy of life in the petri dish. When the condition is correct, they will be able to differentiate into the cell types we have in our body. By histology, it's about 220 different cell types. I think the idea, you got the idea. And in most recent terms, especially since, so the first successful isolation of the embryonic stem cell from mouse was accomplished in 1981. And then it took about 17 years for the same feat to be accomplished for human. So when Jim Thompson from Wisconsin isolated the human version of this, and there's a lot of interest because maybe in our hands, human hands, the ability to produce virtually all the cell types we needed, if say sometime in the future we need them. So the idea of regenerative medicine comes. There are many problems with this one too, obviously. There's no real successful regenerative medicine except bone marrow transplantation has been used over the last 40 years or so very successfully. A singular hematopoietic stem cell would be able to repopulate the entire immune system or the blood system of an individual. So that sort of gives you the prototype of the regenerative medicine. If you can do it in the blood, obviously you should be able to do that with everything, maybe the brain too. So there's a lot of ways we can think about this. But there are many problems. One of the problems is once we became individual like us, we don't have our blastocysts anymore, okay? If you use some other people's blastocysts, and then the immune system will be able to recognize that and then maybe destroy that cells whenever you implant it to that. So there's the idea of patient-specific pluripotent stem cells, namely, can we make customized customized pluripotent cells? That's the one shown in this dish right here. So the science has been, so once we become this is no longer available. That's why I'm basically telling you, okay? So the idea of turning the clock back towards the pluripotent stage has been around for a while. In the 50s of the last century, and John Gurdon, among a few, being able to use a frog system, Xenopus, the frog that you see, and put a differentiated nuclei into egg, and then be able to regenerate a new clone frog. And the same thing happened with Dolly in 1997. Okay, so that's the first time it's happened in mammal. So this is the first indication that although the adult nuclei have been differentiated, and somehow with the magic of the oocyte, the clock can be turned back in terms of cell fate. Of course, and that has never been accomplished for humans so far to this day. So it's been very difficult for the cloning process. And not to mention the ethical problem associated with it. There's another way you can turn back the cell fate clock by fuse a pluripotent stem cell with the somatic cell isolated from an individual. So in that case, the pluripotency, the property of pluripotency is dominant over the somatic one. So therefore, whatever the factors embedded within the pluripotent stem cell can rewrite the code for the somatic cell and turn that into a pluripotent state. So the most remarkable achievement over the last couple of years has been the singular discovery that, in a very rational way, these fibroblasts, as well as few other cell types, can be converted back to the pluripotent state with just a handful of transcription factors. Okay. So for those of you in the audience, physicists, transcription factors are the master regulators of cell fate. So they were able to be used to convert and differentiate cells back to the original pluripotent state. So the scientists, there are many people really trying to do this. The one succeeded doing that is Professor Yamanaka with his postdoc Takahashi. So that has been the sort of most excitement for the last couple of years in the stem cell field, and it's also the subject of this particular session. So that's my basic brief introduction. So what I'm going to tell you is what happened after 2006. Worldwide there's effort to obviously understand the mechanism. How can 4 transcription factors turn back the clock of differentiation? Secondly, the technology that existed at that time, very primitive, very inefficient. It's just like the chip industry. Once we're able to put transistors on the silicon wafer, and there have been a continuous drive to improve that technology, I think the same thing is going to happen with IPS technology. And lastly, obviously, we would like to leverage this technology to help patients, not only for cell-based therapy, as I mentioned previously, and perhaps to generate a library of patient-specific stem cells. And those cells will be far superior than any models we're using for drug discovery. Just imagine we would have human version of a particular patient, the embryonic stem cell of a particular patient, and we knew the history of that particular patient suffering from heart disease, cancer. It's like you're having a DVD, of life of that particular individual. You can play back again and again on our experimental system. You can stress them any way you want and try to reproduce the phenotype. Of course, once you reproduce the phenotype, then you will be able to find ways to find, say, a drug, antibody, or any other technology to alleviate that particular defect. So those are the basic roadmaps as we envision it in 2007. In my own laboratory, I just take probably 1 or 2 minutes to work down a few things in that. Many of the reprogramming processes start with fibroblast. So one of the findings we had in our laboratory, and we have identified an intermediate during the reprogramming process. That is that there's a mesenchyme to epithelial transition in the intermediate step before they became pluripotent. So this is a quite interesting process. Many of you, a few of you are developmental biologists, you know the reverse is the epithelial to mesenchyme transition, one of the key processes during early development, which generate the entire mesenchyme. So along the way, we discovered that the TGF-beta is really the major blocker for the reprogramming process. So anyway, if you have TGF-beta, you're going to have a tough time to reprogram fibroblasts because the keep down fibroblasts become fibroblasts. The second one is we discovered by simply adding vitamin C to the culture media, we were able to enhance the reprogramming process dramatically. And most recently, we discovered that vitamin C actually activate one of the histone demethylases, GHDM1A and 1B, and in conjunction, they will be able to suppress the senescence program or associated reprogramming, and then allow the reprogramming process to proceed. So the future, as Professor Qizhu is probably going to outline you more elegantly, is that any one of us, in our laboratory, our favorite cell source is actually from the urine. And it turns out every day your kidney is shedding reasonably healthy cells called podocytes. And they are actually epithelial cells. My first discovery of the mechanism is the fibroblasts become epithelial cells become pluripotent stem cells. And then we can start with the epithelial cells, and now they can be converted to pluripotent cells in a very robust fashion. So it's very, very good. And then the whole field is now improving this technology, also try to be able to convert this into any functional cells, such as like a neuron. People suffering from a spinal cord injury, they will probably need functional neuron to replace them. And then perhaps the whole cycle can be completed. So there's a tremendous amount of effort going on in China, of course. And Professor Qizhu is a leader in the field. I think he's going to tell us what's his most exciting thing happening in his lab, and perhaps as well as what the future really holds for this exciting technology. So with that, I'll turn the podium to Professor Qizhu.